Hello, everyone. Can you hear me?
Yes.
Great. Yeah. So, welcome to day 2 of this IQ virtual workshop on machine intelligence for quantitative modeling and drug discovery and development. I hope you're able to attend day 1 as well, where we had I don't need to show my screen. I hope you're able to attend day 1 as well, where we had 3 wonderful sessions and presentations from the FDA, academia, and industry. And but if you were not able to attend, we will share both the recordings and the slides on a public link after the workshop. So hope today we're able to have, I think we have quite a number of great presentations today, and today's focus will be on drug discovery. AI and machine learning has been applied in this field in terms of candidate selection and understanding safety endpoints for quite a while. And so I think these talks will highlight that, but also go into new advancements in the field. So without further ado, I would like to introduce our first speaker, who will be giving a talk on in vivo QSAR ADME tools to augment drug discovery efforts, John Sanders. John Sanders received a BA in chemistry from Colgate University and completed a PhD in physical chemistry at the University of Illinois at Urbana-Champaign. At UC, John worked with Eric Oldfield and used molecular modeling to characterize and design novel biophosphonates for their potential use as anti-parasitic agents. He joined Merck in 2006 as a computational chemist in West Point, Pennsylvania, where he primarily worked on neuroscience and infectious disease discovery programs. In recent years, John has been a champion of the prospective application of machine learning for decision-making in various discovery contexts, particularly in the area of ADME properties. And beginning in late 2020, he has also partnered with colleagues in the development and worked to bridge the computational efforts in the discovery and development organizations. So I'll let you take over now, John.
Yeah, thanks so much for the introduction, Nick. I'd also like to thank the organizers for the opportunity to participate in this workshop. This is an area I'm really excited about. It's a place where I've spent a lot of time and energy over the years, and I think it's really cool to be able to be here to, participate in a panel and engage with the attendees through the panel and Q&A discussion later. So what I'd like to do is start with just a sort of brief description about how we think about compound maturation in our discovery programs at Merck, and I'm sure much more widely in our business. Obviously we think about trying to synthesize the best molecules and collecting information in an increasingly costly way about those compounds as they progress towards candidate workup. And one of the things that I think we've done at Merck is really try to bring machine learning tools to the table in order to try to inform on which molecules are the best ones to synthesize, but also how to progress those compounds judiciously through the maturation process. And so in particular, I think we've done a pretty good job of bringing models to the table for every one of these elements that we think are important for the prediction of human dose. So when we talk about potency, we can use both physics-based tools, but also machine learning tools to estimate potencies before we go in the lab and make a compound. And as we collect information and advance onto more costly assays, we have models for things like DMPK properties, for instance, mean residence time in rats and dogs. When we talk about more complex phenomena like bioavailability, we have models like permeability and solubility, which we think help identify the important contributors to having good bioavailability. And as you move towards PD and safety assays, we'll consider things like toxicology models or distributional models like PGP or BCRP efflux to try to make sure that we're advancing the best molecules possible into those studies. And I think that the influence of machine learning has grown over the last decade, certainly. And what I'd like to do with the rest of my time is provide a couple of examples of that and maybe pontificate a little bit about why I think we've been more successful more recently than we have in the past. So when I arrived at Merck in 2006, we already had a number of models for different kinds of ADME properties. We've added to those models over the years. They've certainly gotten better with additional data and additional techniques and descriptors, but I think there's a handful of things that we've done that have really helped us move the needle in terms of adoption and uptake. One of them is really simple, and it's just starting to use our continuous models more categorically. And I think one of the reasons why this has been helpful is because when we stop really emphasizing things like R squared or focusing conversations on how much error there is in a given result compared to its predicted value, I think we've changed the conversation and make it much more probability focused. And I want to use the MRT model as an example of that. So on the right-hand side here, I have a plot bar chart showing bins of mean residence time in a rat. So very short, less than 3/4 of an hour, longer, greater than an hour and a half in this intermediate range. And what this bar chart represents are the prospective predictions made prior to measurement in an animal. Each bar is colored by what the experimental outcome from the in vivo study actually was. And so instead of focusing on whether or not a molecule is predicted to have an MRT of 1.6 hours or 4.2 hours, we're instead really focusing on whether or not the property is sufficient to move the project forward. And so if the team is really interested in a compound that has an MRT greater than an hour and a half, rather than really focusing on the numerical precision of the prediction, we're more interested in whether or not the classification is essentially correct. And so in this case, 70% of the time when we predict that a compound is going to have a rat MRT greater than an hour and a half, that is confirmed experimentally. And on the flip side, when we predict that something is going to have a short MRT, less than 3/4 of an hour, that's confirmed about 60% of the time. And so by focusing instead on trying to select which compounds are most compatible with our needs, I think we've moved the needle and helped teams start to accept these tools a lot more. Another thing that I think we've done more recently is really starting to focus on opportunities for usage within project teams. And I think that has two real important components. One has to do with how much we're actually using the tools, and the other is which tools are really the most appropriate tools for the needs of a given team. And you could think about applying all the models that we have to all projects, but the reality is that some teams are struggling with certain aspects of the definitions of good, for instance, QSAR activation or permeability. And what we try to do is really focus on the models and the thresholds that a prediction might have that define if a compound is good or bad. And so by focusing our energy, I think that's helped us drive to greater degrees of acceptance in our project teams. And lastly, I think one of the things we've done is improve our ability to talk about the benefits of improving compound quality and how we progress our molecules. And by emphasizing the opportunity cost of the organization, for instance, if someone chooses to make a molecule that looks like it won't really progress very successfully, that's coming at the expense of a molecule that could have been better. And I think by also showing in some cases the actual cost of some of these assays, we've driven more interest and acceptance in trying to put the best molecules possible into those more costly experiments. And so overall, I think that we're really moving towards a future where we are able to focus our synthetic and testing resources on the best molecular designs or physical compounds that are consistent with program needs. So I mentioned thinking about the statistics of usage. And one of the ways we've made progress in identifying when we can do better is by using our virtual design database. Essentially, every time a team makes a prediction for a molecule, a compound goes into a database and it has a virtual registration date that's associated with it. Once a compound advances to being a physical molecule that's in a bottle, it of course is going to have a physical registration date. And we can compare those two dates and ask the question, was this molecule evaluated in an in silico fashion prior to its synthesis? And what that looks like is this plot that I have in the bottom left here. So I'm showing you a panel of individual bar charts. In this case, this is a project, and we're looking at the date range from January 2020 in the top left, down to April 2021 in the bottom right. In each panel, a bar represents the compounds made by an individual chemist on this project team. And the compounds that are shown in blue are compounds where the virtual registration date precedes the synthetic registration date. So these are cases where we believe that a team member has gone in and made some sort of in silico evaluation for these compounds. What I think is noteworthy about this set of individual months is that somewhere around the middle of 2020, like August, September, you can see that there's sort of an inflection where a lot of the red is replaced by blue. And I think this really marks a point in time where this project team started to consider using those models a lot more. That doesn't necessarily mean, of course, that just because someone is generating an in silico value for a compound, that number, that piece of information is being used to the greatest extent possible to influence decisions. And so I think we need to consider something different in order to really try to understand the value that these models bring to our teams. For that, I think one thing that we can do is just consider the distributions of compounds. So for molecules that are synthesized but never progress to, that are designed but never progress to actual synthesis, you might expect a much broader distribution of fitness. So if we imagine, for instance, this plot that I've just animated at the lower right is something like solubility, of course, a team is going to be capable of designing some molecules that are predicted to have good solubility, many that are predicted to have mediocre, and some that are predicted to have terrible solubility. If we're using these models really effectively, what we should see is a shift in the distribution of properties, such that for the molecules that are actually synthesized, we would expect there to be a differentiation in the properties. Hopefully, we would be selecting the compounds that have the best fitness and focusing our synthetic efforts on those. And of course, there's going to be a tail for things that are anticipated to be inferior, but there's really good reason to do that. So considering permeability, for instance, maybe a team is struggling with permeability, they want to improve it, they want to do so by introducing an intramolecular hydrogen bond. If the QSAR model is unable to really anticipate the effect of that, then it's going to predict that the compound is impermeable. But having a really good rationale for why that design is appropriate to move forward with should drive a molecule forward. And so there is good reason to expect there to be a long tail for undesired properties. In practice, I think we can see examples of where those distributions really start to diverge in a meaningful way. So I have an example of a project. It's a neuroscience program. This team is using our PGP model to prioritize molecules for synthesis. On the left-hand side, I'm showing a distribution of predicted QSAR rat PGP efflux ratios for designed molecules that were not synthesized. You can see that the most probable prediction is for molecules to be substrates with an efflux ratio greater than 4. However, when we look at what the team has actually gone in the lab and synthesized, the predominant fraction of these molecules are predicted not to be PGP substrates with efflux ratios less than 2. It's also true that based on the coloring here, you can see that the majority of molecules were evaluated in silico prior to synthesis. And I think that really helps exemplify that it looks like this team is making predictions before they go in the lab, and they are focusing their synthetic resources on the compounds that are predicted to be most compatible with what this team needs, which is non-substrates so that they can have CNS penetration. So that's the synthetic aspects. But I think we can also use models to really define which molecules progress once we put them in a bottle. So now what I'm showing is a corrected potency metric on the x-axis. In this case, a greater number is superior. So we want to be greater than 6. And you can see on the left-hand side, I'm coloring these by their QSAR PGP efflux ratios. So the majority of molecules are predicted not to be substrates. And when we look at which molecules are advancing into our PGP efflux in vitro assays, you can see we're advancing our really potent compounds. And here what I'm showing is the fact that the experimentally determined PGP values that we get from these prospective predictions are predominantly skewed towards the non-substrate region of property space. So I think this exemplifies a team that is making predictions on a large number of candidate designs. They're picking the best ones for synthesis, and then they are moving those predicted to be high quality compounds into that in vitro assay. And we are actually demonstrating that those compounds are translating to be appropriate for this team's needs. We can also do this with more complex phenomena like mean residence time in a rat. So this is an older project that was interested in trying to improve their MRTs. They were struggling to do so. And the team realized that the model offered enough enrichment to start to identify more promising compounds before they were synthesized. So the chemistry lead decided to ask the team chemists to come to design meetings with the molecular structures of interest, but also with predicted MRT values. and they agreed to avoid designs where MRTs were shorter than 3/4 of an hour. On the left-hand side of the plot, what you can see is two distributions superimposed. So in blue are compounds that the team actually went and physically made, and red are molecules that were designed but not made. And what you see in this case is actually a bias for molecules being made which have predicted MRTs that are insufficient for their needs. Once they implemented these design criteria, what they were able to do when they got to the first half of 2019 is really shift the distribution of the molecules they're making towards the longer MRT space. So they've eliminated a lot of the molecules that are predicted at very short MRT. I didn't label it, unfortunately, but the right-hand side of this plot is illustrating what the in vivo measurements were subsequent to synthesis. So by focusing on, interesting, back to full screen, I hope, for everybody. By focusing the chemistry resources on compounds that have acceptable MRTs, what the team did in 2019 is actually increase the fraction of compounds that have MRTs greater than an hour and a half, and measured MRT greater than an hour and a half. And I think what the data on the right shows, the team was able to improve in 2017 and into 2018 the fraction of compounds having those longer MRTs, but they kind of plateaued. And when they recognized that they could use in silico tools to really help focus synthetic efforts on more likely to be successful compounds, that did actually translate to an uptick in compounds that have these right properties. So I think both of these examples illustrate our ability to really steer our synthetic resources the right way, and for that to translate to more successful molecules in later assays. I think there's an opportunity to accelerate projects, though, not just to pick the right molecules. And so if we think about using QSAR models to make the right molecules, to pick the right ones for potency or for PGP or cassette studies and so on, ideally, if we're looking at a compound has not been made yet, it has an undesirable efflux ratio or predicted MRT, that's a compound that should die. It should not move forward. It would be very inefficient to make those molecules, or to make many of them, I should say, because I obviously give an example in the case of permeability, when you may have a really good reason to override a QSAR prediction. But I'm really talking about when the evidence points towards the design being undesirable here. When you have a molecule that falls into sort of that indeterminate range, where there's a chance that it's actually quite good or quite poor, that's a molecule where your uncertainties really dictate that you have to go and measure them. So you would need to take that compound, put it in a potency assay, wait until you have the right results, move it forward to a PGP assay, wait until you know that it's sufficient before you put it in an in vivo study, and so on. But what if you have a molecule that has a pretty good prediction of something like PGP efflux, and you feel really confident about that chemical series for your program, and for that prediction to generate a good outcome? Maybe you can skip the PGP assay and go straight to something that's deeper in your compound maturation process. For instance, maybe you go straight to a rat cassette. And what this represents is the opportunity to accelerate the most promising compounds and put them into the assay where they're likely to fail first. Of course, you could do the same thing from the point of view of focusing on compounds with good MRT, but ideally what you would be doing is accelerating the compounds that are aligned with all of the needs of the program and advancing them as quickly as possible. Now, this may seem kind of far-fetched because you could imagine there may be a lot of compounds that meet all of the right thresholds, but perhaps in those cases what you can do is be more aggressive in using higher thresholds and cutoffs for a good prediction to be considered for advancement, such that there's only a small number of compounds that you're accelerating in this fashion, but what this would allow you to do is ultimately complete more design cycles in the same fixed time period. So I'm going to change gears for a minute because I think that there's also opportunities for us to use machine learning in development. I'm going to use a chemistry example. I think that that chemistry example will also apply in the discovery space. So one of the things that we often do is we use catalysis kits from Use Automation at Merck to try to identify, for instance, really good ligands and catalysts for the chemistry that we want to do as part of defining a good manufacturing process. And I'll use this pyridine oxazoline example. This is a ligand class that's useful in a handful of kinds of catalysis. These are combined from a couple of simple building blocks, easily prepared. And the question that we are interested in is whether or not we could use this kind of a ligand class with some high throughput data in an iterative process to get to better and better molecules more quickly. So what this team did was, over a series of successive rounds, they took a set of molecules, they generated some data. In this case, we're measuring %EE. They then used that data to train a model, which they used to then predict some additional molecules that they were interested in. They outsourced the synthesis of those, they tested those, and then they reiterated on this process. And so in the first round of 32 ligands, Q² wasn't great. You can see that the enrichment plot at the bottom indicates that at least the best molecule showed up on the right-hand side of the bar chart, but not really great performance. However, after another round or two, we can start to see the enrichments improving. And after round 6, not only are we able to successfully predict the compounds that give the greatest %EE, but we're also able to predict into that higher %EE space. And so what I think this started to show us was that if we're really careful about how we design these plates and how we iterate, we may be able to accelerate this by shortening the number of rounds that we actually use. So the idea is start to use clustering in order to make the best possible plates to maximally enable our machine learning process. So if, in this case, what the team was able to do is enumerate a large number of possible pi-ox ligands. Some were chemistry informed, some were from just virtual database enumeration, some came from the literature. And if you cluster those into a subset of clusters and pick some diverse centroids, that gives us the ability, we think, to start to design really carefully curated plates that offer chemical diversity that hopefully position us to accelerate that machine learning and iterative process to help us discover the best possible chemistry as fast as possible. So I think that this is a nice example that we hope to see a lot more of in the future. And obviously, I think I've shared a few some examples of in vitro and vivo and also chemistry-based success of machine learning. So how do we see the world right now? I think we are using machine learning pretty effectively to select the highest quality designs, to advance molecules effectively once they've been synthesized, and to optimize chemistry. And I think recently, because of the successes that we're starting to see, we have decided to invest further. So for instance, we're starting to do things like increase our prediction capacity. We recognize that we'd like to make more predictions on more molecules, and we need to drive our infrastructure to keep up with the pace that our community demands. We're starting to use generative models, and we believe that that's going to unlock areas of chemical space that we haven't imagined before, and having access to good quality predictions to help steer our synthetic efforts in the right way is really important to building confidence in those models and the compounds that they output. We're improving our retrosynthetic methods, and I think we're figuring out how to maximally impact our organization with those approaches. We're starting to build more toxicology models, and we're figuring out how to use them effectively to guide decisions and discovery, but also chemistry. And I think we're starting to gain experience with machine potentials based on physics-based modeling, for instance, energies of conformations. And I think longer term, what we anticipate is that the organization's machine learning fluency is going to have to increase. And I think that the modeling field is going to evolve as well. You know, I think what historically in computational chemistry, which is where I consider myself to sit, is based on physics-based modeling, but we're going to be moving towards individuals who have much more machine learning backgrounds, such that these folks can bring these tools to their teams with increasing competency, but also frequency. And so what I'd like to do lastly is just to acknowledge a number of folks who've contributed to this work over the years. I think that people have contributed both directly and indirectly to our thinking, and I'd just like to thank all of them. And so I think at this point, I will stop sharing and hand it back to Nick.
Well, thank you, John. That was really a great presentation. And to the audience, we're at 216 attendees, which is great. If you've got any questions, please enter them in the section there on the, at least for me, on top right. John, again, thanks, and we'll come back to you in the round table, and we can answer additional questions. I'd like to now introduce our next speaker, Rich. And Rich, if you can get on camera. Rich actually is a colleague of mine, so we work on a lot of joint projects, so it's great to see you here, Rich. By way of introduction, he got his PhD with David Baker, which is, of course, he's a very well-known field. And then after a couple of detours, he became professor at NYU. And then not too long ago, he started Prescience. And the unique thing about Prescience is we talk a lot about AI and ML for small molecules, but that really focus on the antibody. And in a very short period of time, they actually achieved so much success that, of course, Genentech bought Rich. And now Rich and I are partners in crime, so to speak, work on a lot of joint projects and making great progress. So Rich, take it from here. Thank you.
Thanks a lot, Marcel. Yeah, it's been absolutely wonderful to be part of the GRED of Genentech and the Roche family. I'll take the next slide, please. Today, I'm going to set up the conceptual framework and then some of our machine learning approaches to how we do machine learning aimed at large molecule drug discovery. I'll try to emphasize a few of the things that we're doing that we do, having talked quite a bit with Marcel and people in SMDD. I'll try to emphasize things that I think are cross-cutting and sort of highlight things that might be special to LMDD and some of the advantages of that system. Overall, we have a set of objectives and a set of approaches to this problem that have been developed in close collaboration with Arvind Rajpal, who is Vice President of LMDD at GRED and Genentech. We also have a great debt of gratitude to Yan Wu, who's done a lot to gear up systems to test at scale. But in general, when we sat down and looked at our capabilities, we really have kind of two separate approaches. One is our set of approaches to take things that we have already, molecules with liabilities, and molecules that are known binders, maybe very early stages, so they don't have developabilities, but they haven't really had a lot of developability work performed on them. We have a set of approaches to take those molecules and move them forward, fix them, and improve them. And here, the antibody, the Fab, VHH, the number of options, the amount of sequence space you have, I think we're very optimistic that we'll be able to make fully automatic methods that solve these developability and optimization problems every time, with a few iterations that are small, a plate or two at the most. Direct design is the ultimate goal. This is the thing that is a little bit more of a leap for both us and for our experimental colleagues, and that's the hope that one day we can take repertoires that come from either computational methods or display technologies or synthetic methods, but take those repertoires and directly mine them, perform machine learning to directly design to a selected epitope. And so here the idea is, instead of taking things with different epitopes and different activities, therefore, we would actually let the biology drive epitope selection and then do direct design. And so our aim split into these two things. And you can think of really this as also being realistic in the sense that Genentech is also already very, very good at designing antibodies. And so the optimization aims are a way that we can have fun immediately, and the direct design aims are the thing that we'll build towards and continue to build towards in the future. So next slide, please. So before I start, I'll just kind of give a little history. We had a very long research program, 20 years for me and closer to 15 for Kyung Young, aimed at structure, prediction, and design. I was involved in the Rosetta community and other efforts there. Many of those codes have been licensed by pharma for many years. Kyung Young Cho, the inventor or one of the co-inventors of the attention mechanism, had been looking at ways to make machine learning properly generative. So instead of classifying, is this good or bad, he's very keen on methods that can actually generate new things. And this is, of course, a very active topic. We came together, and Vladimir Grigorovich, who comes from machine learning and hails from Imperial and sort of the London School of Machine Learning, he joined us. We founded Prescient. We were able to recruit a lot of structure talent, in Andy Watkins, and some machine learning talent, absolutely brilliant machine learning talent in Dan Berenberg and Steve Raw. Well, once we had that up, we built prototypes, we talked to Genentech, and we realized we had something that worked a lot sooner than we expected. Joining Genentech was the luckiest, best thing that could have happened to us, in the sense that now we have the ability to, if I could get the next slide, really implement a nice integration with the laboratory and the ability to test designs, and that's essential for machine learning. I'll get into the details of these things to some degree in the next few minutes, to the degree that I'm able. But here the idea is we have something called the manifold sampler. And again, I think diving into some of the more technical details here is actually a little bit necessary to just to illustrate our approach. And so I apologize, but I will dig in a little bit into some of the algorithmic details here. So we have a manifold sampler, and here the idea is this is a machinery that's seen every antibody and every protein, and so it knows how to generate sequences that are protein-like, and we could put that in air quotes because there's a lot of different types of large molecules, a lot of modifications, a lot of ways they fold, a lot of ways they're dynamic. And so this is something that is an active area of research. But we have this manifold, which is a way of, with a machine learning, changing a sequence or generating a sequence that has foldability, local structure, and other properties that are proper. In that system, we have oracles which predict properties, but they predict properties without needing to leave the machine learning space, which means many of our property predictors can guide machine learning in a way that's much more efficient than sampling from your machine learning or your experimental protocols, judging it, and then putting it back in. Instead, many of the methods that we have can actually provide direction finding deep in the machine learning space, which is extremely efficient. All of these things feed into an experimental design system, an active learning framework, is maybe the easier way to say that, which helps us to algorithmically select the proteins, the antibodies that we're interested in. We then work very closely with LMDD to call those, to put some humans in the loop to look at those structures and those sequences. We finally assay a select number, typically a plate per target, and then we provide feedback to the model in the way of augmented training, and actually more importantly, augmented updates to the active learning framework. And so this is something, again, that we've done with a deep integration with large molecule drug discovery at Genentech. Most of our project meetings are half people in LMDD and half people in Prescient. And so there is a lot of influence from the experimental side in terms of their selection of properties to optimize, ways to call and fix data sets for training, and all these sorts of things. So this is a very tight collaboration. Can I get the next slide? All right, and so I'll go into next slide, a little bit about how we, what we mean by manifold sampler. And again, there are many varieties now of these types of methods, and there's a very active research program at Genentech, not just at Prescient, but in many places to perform these types of machine learning operations and build these types of models. But much of what we do relies on something called the manifold assumption. And here the idea is that real world high dimensional data lies on a low dimensional manifold. And this is a problem, or a statement that I think wouldn't have been surprising 50 years ago. And there's been a lot of really recent amazing work to operationalize this assumption into some really powerful computational methods. And so if we look at some of the early works with manifold machine learning, self-supervised machine learning applied to vision, the idea is that there is essentially a number 4, and that number 4 has all kinds of operations, noise, rotations, translations, and those are actually much easier to represent than trying to represent or remember every single 4 that's ever existed in every single context. Now with proteins, I think we do know that the history of protein folding history of modeling molecular interactions with proteins has had a fair amount of, or has been dominated by physical models, but it's also had an equal amount of extraordinarily clever ways of making sure that our proposals, our samples, the things that we do to proteins are remaining protein-like. And so some of the earliest examples were from David Eisenberg's lab and from the Baker lab, where fragments of local structure were assembled to predict larger protein structures. And here, what we're doing is obviously substantially more advanced, but the idea is that when we propose mutations, we propose them 15 at a time, and those 15 mutations are better than the majority of single mutations, right? And so that's a very arrogant, tough statement, but we have a lot of empirical evidence that the technologies for molecular manifolds are actually holding up to those types of statements. Can I see the next slide, please? Here's again, this is more, I apologize maybe for a large molecule crowd, but there are a lot of distinctions in these methods. And again, we have many different varieties that we're experimenting with at Genentech. But one key distinction that you can apply to these self-supervised methods is the way in which they generate the output from the latent space. So when you've encoded a protein or a small molecule into a manifold and then you spit it back out again, do you start with one atom or one amino acid and then have a sequence which generates that, or do you have some way of predicting the size of the system and then outputting the system in a single operation? And the early days of Prescient, we were very keen on non-autoregressive models, these models where you output the sequence because when coupled with a good length predictor and some other constraints, oracles, things like that, we found that we could actually make methods that could walk between folds, that could optimize Rosetta's scoring function much faster than Monte Carlo. And most importantly, we could change the length of things. And that is very key to, I think, optimizing binding, the ability to change the overall size of the system as you're generating the system. Now, for antibodies, that entails changing the length of the CDR3 loop. And a non-autoregressive model is perhaps a nice fit for that. That's not to say that with autoregressive models, you couldn't find many heuristics to wrap them in that could do the same thing. But this is a key distinction to keep in mind as you shop for these types of models. And so I wanted to just stick on this for a minute. Next slide, please. So for our systems, we usually have three components. We have the encoder, we have the decoder, and then we have the length predictor and the function classifiers. That are things that are essentially generative models or oracles that are embedded in the overall space of encoding and decoding that can provide us direction finders. And so here, the length predictor is something that's required because this early model is a non-autoregressive model and had showed great power in perturbing the CDR3. Next slide. Next slide, please. All right, so now next slide, please. Now I'll jump in. I think I'm going to run out of time relatively soon, so I'll jump into some of our applications. But first, I wanted to hang on this slide for long enough for people to see that we have, for different of the operations that we have to perform, we have been lucky to be able to publish some of these things. So using some of these methods to predict, function, and annotate vast swaths of microbiome sequence data and unknown genomes and things like that. That was one of Vladimir's earlier papers. We then sort of turned that in the opposite direction, and we have a paper on function-guided protein design where you can dial in the enzymatic functions, cofactors, various features of function, and automatically generate sequences there. And of course, those can be thought of as final designs, or they can be thought of as samples that you feed to downstream processes. We have some other things that are more sequence-based, including fast search and a method that's quite tuned to cases where you're optimizing molecules, antibodies, for example, where you know there are regions that shouldn't be changed, how do you deal with those boundary issues, and how do you generate things that fold substantially more efficiently than if you didn't properly address those boundary issues. So these are a couple of different papers that you can dig into that actually have a fair amount of detail included. Can I look at the next slide? All right, so now I'll go through just kind of how the rubber meets the road, so to speak. So when we landed at Genentech, immediately we got to work on developability. This is a problem that is extraordinarily interesting because many of the properties that we think of as developability properties for, well, either for large or small molecules, but especially for proteins, they often have really different underlying structure, right? So we have things like affinity, viscosity, and expression, which might be very clear derivatives of the structure, right? The best, in my mind, the best way to approach those problems is to look at the three-dimensional structure or the structure of the interface and start going crazy, right? There are other things like polyreactivity and immunogenicity where you have to sort of imagine an ensemble of interactions or a whole system in the case of immunogenicity. And these things are very well characterized, and they have really wonderful proxy assays. But in general, we have very different kinds of properties that have to simultaneously interact. Here, the ultimate goal is to put them in as oracles into the latent space so that we can extremely efficiently say that anything that we generate satisfies these oracles. There's a great amount of data that goes into these models, and we've had a huge amount of support from Genentech Informatics, so we have to thank them for that. And then testing these models also implies a fair amount of effort. But the key principle here is that we now have upwards of 5 properties that are integrated as oracles simultaneously. And so when we test a molecule, we are not generating a large batch, testing a single property, filtering. We're not suffering from this multiplicative serial dilution that I think is present in many drug discovery workflows. And so we do think that we're making progress towards integrating these different developability properties and solving the serial dilution problem. Next slide. The other thing that we're very keen on is looking at the vast quantity of data that exists at Genentech and Roche, but also the vast quantity of capability involved or implied by having the animal facilities that we have, the ability to immunize a wide variety of different types of animals. Exploring the epitope diversity and the structure of repertoires is something that will remain relevant for some time, and is extraordinarily powerful. So we've been working very closely with a number of people at Genentech to work on these methods. And these are methods that are essentially machine learning methods for finding great feature spaces, clustering those spaces, and then coming up with diverse samples from those spaces. And I think we saw a little bit of that in the last talk. That was quite nice. So I won't focus too much on this. Next slide, please. So the ultimate golden goose would be direct design by de novo design of affinity. And here I could say that we made quite a bit of progress with both machine learning methods and hybrid structure methods. We have a paper that we're putting together now that is focused on a combination of our machine learning methods and Rosetta. I won't steal Simon's thunder too much. I want to let that progress, but I can say that key to that effort is the representation. And there's been a lot of really amazing work lately on graph neural network representations and related representations. There's also been a lot of really amazing work on ways of encoding molecules in machine learning that are invariant upon rotation and translation, and that are perhaps very well suited to modeling interfaces. And so we have a lot of prior work that we can use to do this. I would say just one thing to point out, there are many different companies and places and academic labs that say they've solved the de novo affinity design. It really pays to read the fine print in those papers. Usually what they mean is if I do a display technology-based experiment, and I get 10 to the 9 things, and I get 20 binders. I use the computer to optimize those and design another display library, and then after 3 iterations of that, I have a binder. To me, that's not exactly de novo affinity design solved. That's more of a really awesome way of designing display libraries, which again, we would love to use those types of methods. But here the goal is to make a plate and to get a nanomolar or sub-nanomolar binder reliably. Next slide, please. All right, so I'm relatively new to pharma, and so completely chickened out in terms of putting any data in this slide. But I can tell you that we have applied our sort of several of these sub projects, including de novo, less, and developability for all of these things, to 2 very, very publishable targets. So one where it's been off patent in several different ways, and one that's, I think, just a component of egg white. So we have OVA and HER as targets that we can publish on, and we were hoping to write up a lot of the details that I didn't talk about today in the next few months. We have 6 hot targets and 4, I guess, medium temperature targets, and we are every 3 weeks making another round of designs for those. And so active learning, this is the thing that is one of the key results that I'd like to talk to you about in more detail, but it'd be a whole talk in and of itself. Our framework for making decisions in this multi-parameter space is working, and we actually have decision making based on algorithms that's led now its 3rd iteration of designs. And I have to say that was extraordinarily frightening to me. And we didn't, it might not have been as frightening to our experimental colleagues, but maybe that's just because the whole prescient adventure has been really frightening the whole time. But either way, we took a chance, and the active learning iteration seemed to work. The developability, optimization of the developability parameters for 5 of 6 targets looks really, really nice. We have some problems with the 6th target. Another key thing with these results is that the methods have been pushing the number of mutations or the edit distance to any given seed to the point where these loops are being completely remodeled, and we're making upwards of 50 to 65 mutations per design, and still achieving high expression, good yield, good binding, and other developability parameters. And so these are much more aggressive designs than you typically see in LMDD. And it's you know, a little bit of an adventure still, but it looks like things are working quite well. Our early readouts from de novo, sort of real de novo, as well as grafting loops onto new things that came out of the computer. This work is looking really quite nice with early readouts, but we have an extraordinary amount of work to continue that project towards this reliable, every plate's got a binder dream. The next slide, please. Yeah, and so I want to thank everyone on the Prescient team. There's too many people to thank, but I do want to especially thank Stephen Ra, who, in addition to being a brilliant co-director of the Frontiers group with Kyung-Young Cho, also really did quite a bit of work to help me prepare some of these images and slides, and has just been brilliant in leading several aspects of our machine learning projects. Jiwon and Natasha, I'd like to thank for a lot of work with various oracles. Jaewon has also been amazing working on some of our de novo work. And then Simon Kilo is our hybrid structure guy. And hopefully we see a nice paper from him this fall on this work. That's it for me. Thanks for your attention. I'll hand it back to Marcel.
Thank you, Rich. Good talk. I don't see any questions from the audience at this point, but they have been coming in after the talk. I guess I had one quick question before we go on to the next talk. You said that on one of your last slides that some new lab techniques were coming out to which would help your active learning methodology. Could you go into more detail about that, or what you meant by that?
Well, so what I mean is that, well, at Genentech, we have a huge investment in genomic technology for target discovery. The earliest part, the part that I have no clue how that works right for now. Well, a lot of those technologies are also extraordinarily useful for scaling up various aspects of affinity or T-cell receptor or other large molecule design problems. A key thing is that often we take these molecules and put them into various formats, and we have really great models. I think we're great models and they're getting better. We also know how to think about the affinity and the binding and the sort of pure biophysical parts of how these molecules work. But exactly how you approach the target protein, and this is whether you have a small molecule or a large molecule, how do you, could we start to differentiate things that will have agonists and antagonists, different sort of activity profiles? This is something that we often do by trial and error. We put these molecules, when we get good binders, into different formats, and we see what happens. And I think a lot of the latest technologies are going to allow us to really dramatically scale up the connection between these biophysical properties and developability properties that I talked about, and the actual drug relevant activities, and the differentiation based on format and geometry and things like that. So that's one class of essentially genomics experiments that is ongoing at Genentech, and I think is going to be really exciting to feed back into these models. The other thing is just looking at pure affinity. There has been just a tremendous amount of progress, even in the last year. We think of next generation sequencing as this thing that just works, but it continues to sort of beat Moore's law. They haven't stopped innovating there. And so the ability to collect affinity data, to scale, to the ability to collect affinity data convolved with other key developability parameters, to put other filters in the loop there when we sort or when we put them into cellular formats to express. This is all progressing at a pretty fast rate as well. So that's what I imagine there. There's also a lot of new machine learning technologies evolving, but in that particular point on that slide, I was referring to just the amazing amount of innovation from genomics that will give us a huge boost to our training sets.
Okay, thanks. And yeah, maybe we can discuss that more during the panel discussion. But I think it is time to move on to our next talk by Nigel Green. He's been kind enough to show up, even though he's been a little bit under the weather this week. Nigel Green is a senior director in imaging and data analytics within the ClinPharm and Safety Sciences department at AstraZeneca. His research interests include the application of machine learning and artificial intelligence methods to understand the structural basis of drug-induced toxicity, understanding their mechanism from high-content datasets, such as transcriptomics, as well as their translation to clinical populations. Dr. Green received his BS in chemistry and computational sciences, and his PhD in organophosphorus transition metal chemistry from the University of Leeds in the UK. During his career, Dr. Green has published over 60 peer-reviewed articles and book chapters, and has been a member of SOT since 2003, serving as an officer on the computational toxicology specialty section since its inception in 2017, and it is the current president. Okay, I'll let you take over now.
Thanks, Nick, and again, apologies to everybody on the call. I was tested positive for COVID on Tuesday, and so I'm still in the recovery stages, I think. And I'm going to use the excuse that if this is a bit of a rambling presentation, then I'm going to blame it on COVID. But unfortunately, that excuse only works if nobody has actually heard me give a presentation before. So apologies in advance if this is a bit rambling. But I have a lot to get through. I'm going to have to go fairly quickly through some of the slides, because each one of the slides, I think, actually is a summary of an entire talk in its own right. But I do want to try and touch on some of the areas where we're using artificial intelligence and machine learning in toxicology, do that. And so the way I've structured this talk is to really just to touch on, you know, what is the problem we're trying to solve with AI and machine learning, and then talk a little bit, touching on some of the work that John presented earlier on prediction of pharmacokinetics. This is obviously an important topic and certainly plays into the role of toxicology, and I'll come on to that in a minute. But then talk about how safety assessment has really changed in the last sort of 20 years, at least since I've been working in pharma, and the quantity and quality of data has significantly increased in safety assessment. And so thinking about the application of machine learning to help us both understand and interpret some of the data that's being generated is a critical thing. And some of those applications are things like imaging and image-based assays, thinking about digital pathology and whether we could automate diagnosis of sort of tissue slices. But obviously, more recently, with the advent of SEND and the SEND terminology for FDA submissions, we now have some highly curated and structured raw data that could be used in machine learning senses. And so this offers some more and expansive ways of using the in vivo data. And then finally, I'll wrap up with a few conclusions. So what are the challenges that we currently face in drug discovery? Well, I think this doesn't come to a surprise to many people, but most molecules just don't become medicines, right? There's a lot of reasons why these things fail. It can be the fact that we just don't understand disease well enough and that we're not picking the right targets to move forward. It could be that the compound doesn't have the right pharmacokinetics, and so it doesn't get into the body, or it's not there long enough, or it doesn't stay long enough or get to the site of action. It could have some unwanted side effects. It could be some strategic things like we were beaten to the finish line by the competition, for example. And so sometimes strategic decisions are made to stop drug projects because we just wouldn't be able to get there fast enough. And obviously synthesis is another area, and I think that was touched upon earlier in one of the talks around retrosynthetic analysis. It's not just enough to know what drug you want, it's how do you make it, and that can be an important thing as well. So as we go through the drug discovery process, our understanding of therapeutic index, and that's sort of the concentration at which we hope to see efficacy versus the concentration at which we see adverse side effects. That ratio is often uncertain at the early stages, and often when you look at target potency versus an off-target potency, you need a large sort of therapeutic margin to guarantee that as you move through into sort of animal and human studies, that safety margin sort of stays as wide as possible. And that's really what we're trying to accomplish with sort of our understanding here. And it's often a lot easier to decrease a safe dose than it is to increase a dose in the clinic. And so what we want to be able to do is maximize our ability to dose escalate in the clinic so that we can actually test the clinical concept and make sure that our target is actually the right one. I think the biggest phase 2 failure is where you don't know if your target would work if you had the right molecule. That is probably the biggest failure that you can have. But obviously dose makes the poison, and that becomes the issue of being able to dose escalate safely. So efficacy can be seen as a function of both the potency of the molecule and the exposure at which it's given, whereas safety is also a function of the hazard that, you know, what does that molecule do that you don't want it to do, versus the exposure at which it's given. So the exposure component is a critical part of understanding the safety of a molecule as well. And so that's why, you know, we've been focusing quite a lot in toxicology about understanding the exposure at which compounds are given, and what the exposure might be under certain circumstances, because that will often dictate what the safety profile of that molecule will look like. So a lot of our work sort of in the last couple of years has been focused on predicting pharmacokinetics. And to be fair, we had spent a lot of time looking at, in the same way that John presented on earlier, looking at predicting some of the in vitro assays that we run on our early discovery candidates. But what we really care about is what happens in an in vivo circumstance. And so we thought, why not try and just model the in vivo output from sort of a cassette dosing experiment. And so we took about 4,000 compounds that have been in an in vivo rat PK study, used the chemical structure and the in vitro properties where we had them, where we measured them prior to that rodent study, and basically used machine learning to see if we could predict some of the PK profile. I should point out that this work has been published recently in Molecular Pharmaceutics. So if people want to read more about this in more detail, then I'll happily share the reference with anybody who wants. We use graph convolutional neural networks to encode the chemical structure. This is something that's come out of a consortium with MIT, is currently running, that we found to be actually an improvement on most connectivity fingerprint type approaches. They seem to be doing a little bit better in most circumstances. So we're able to use sort of better chemical structure representations. We've also used, as I say, the in vitro ADME data as input features, but where those features are missing, we've used in silico models to predict those. So that it's almost like a transfer learning effect of using a sort of in silico model to predict the in vitro features of that molecule. And what we get out is actually models which are pretty good. And I think this sort of piggybacks on some work that was published back in 2015, where we found that using lower numbers of compounds that have better quality data gives you just as good models as having large amounts of data that is not necessarily of high quality. The data quality is actually one of the bigger drivers over model quality. And so with rat in vivo studies, the compounds are usually purified quite well. We don't typically just take the first batch and run it in a rat straight away. We'll purify and resynthesize just to make sure that we're testing the right thing. And what we find is that we get good quality data coming out of the rat PK experiments, so that the model quality is actually getting close to the experimental variability that we see within that. So as you can see from the table at the bottom, the RMSE, which is sort of, I should point out, these are in log units. So it's it's 0.28 of a log unit, is getting close, or at least within an acceptable tolerance of the experimental variability that we see within the assay. And the same for the fraction absorbed and also the volume of distribution at steady state. And as you can see from the pie charts at the top, that 75% of the predictions that we make for clearance are within twofold of the actual experimental value, which is actually pretty good for an in vivo experiment in itself. So having had some success with rat, we actually thought, well, we actually really care about human, to be perfectly honest. We're not optimizing for rat PK. We want to know what happens in humans. So could we take data from the sort of the FDA summary basis of approval documents, or from the EMA equivalent documents, and use those, the human data, to try and predict whether we could predict the PK profile of a compound in humans. And so using the Elsevier Pharmapendium database, which actually nicely summarizes all of that data for us, that we actually managed to get a data set of about 1,000 unique molecules with about 12 PK parameters that we could start to use in a multitask neural network type approach of modeling. We started out with about 1.2 million records sort of coming out of the database, and through sort of a expert curation process, we whittled that down to about 4,491 unique entries, because obviously compounds are given at different doses in different trials for different reasons. and so you end up with multiple records for the same compound. And so we had to then think about how to normalize across doses within that. In actual fact, what we did in the end was just use dose as one of the features that was input. And so now what we do in the model is that you input both the compound and the dose, and it will predict the PK profile that you might expect to see from a human. Again, this work has been published in Molecular Pharmaceutics, so if people are interested to learn more, then please go to the paper. The reference is actually on this slide at the bottom. But we were reasonably confident that we could actually predict human reasonably well, I should say. I wouldn't say it's the best particular model here. But what we found is that random forest seemed to work better in this case for the machine learning model. We validated it on both external and our own internal clinical data. And moving on to think about sort of the models, we managed to get from this first attempt sort of 3 decent models, which was AUC oral, Cmax oral, and the volume and distribution from IV. To explain the bar graph, the mean predictor is basically saying if you take the average value for that particular property, that particular parameter, how well would that predict for each compound? What would be the root mean square error? And so what you want is a model that predicts better than the mean predictor, and as close to the experimental variability as you can. And that explains why in this particular case, you can see that the model is predicting far better than the mean predictor, and it's getting close to the experimental variability. In the case of clearance for oral, you can see here it's doing slightly better than the mean predictor, but it's nowhere near the experimental variability in the data. And so we didn't think that this was a particularly good model to use. Obviously, data distributions and availability are going to impact on model performance. And so we're looking to think about, to see how we can expand this data set and get better quality data in the future. And so some of the thoughts are around perhaps thinking about using our rat PK model and using that as sort of an input into the human prediction. And in actual fact, that does improve the performance. We've also looked at whether using dog PK would actually help us, at least looking at feature importance within sort of the predictive models. The dog PK seems to have a high contribution to the performance of any model for human. And so I think the sort of using both a combination of rat predicted properties and dog predicted properties would actually enable us to predict human PK in a much better sense. Whether we could use different species and get better performance, I don't know, because we're very much limited on what data is available to us. And obviously rat and dog are the 2 main species that we use, sort of traditionally used for looking at or projecting out for human PK for small molecules. I should point out that we've also looked at whether these models perform well enough for things like PROTACs approaches, and provided that you have in some representation of PROTACs molecules in your training set, they do actually work quite well as general models. In actual fact, we're struggling to get better quality models looking at PROTACs alone, primarily because as you do that, the number of molecules drops significantly, and so you get less diversity and less the model is less able to differentiate across those different sort of different feature spaces. And so actually the general model is outperforming some of our more targeted approaches. So that's about efficacy. I do want to touch on or at least talk about toxicology, seeing as that's why I was asked to come and present. The face of toxicology is changing quite dramatically. As I mentioned, we're using a lot of high throughput multiplexed approaches now, thinking about microphysiological systems. We're using a lot of image-based technologies. We look at multi-omics technologies. So we're generating large amounts of data on the molecules in a number of different assays. And I think data science and machine learning are going to be critical in improving the way that we analyze and use our data in the future. But thinking about discovery toxicology, traditionally we've had a number of in vitro assays that we'll look at. So things like HERG is the classic example of an in vitro assay that we've applied to look at QT prolongation. We look at other ion channels. Obviously, we have cell-based systems for sort of cardiomyocytes and looking at structural damage to cardiovascular tissues, etc. There's been a number of publications around hepatic injury and assays that can be used for that. Obviously, things like the Glugal assay for mitochondrial toxicity, thinking about cytotoxicity, hepatic spheroids, thinking about liver transporters. Genetic toxicity is another area, lots of in silico models for that. And then obviously thinking about other effects of compounds, so things like secondary pharmacology, that's another important one. Things like phospholipidosis is another. There's been a number of papers looking at rules, structural rules for phospholipidosis. We're still, in my opinion, struggling to get beyond sort of that sort of basic lipophilic molecules are going to generally cause phospholipidosis. And so, your first culprit when you see phospholipidosis is to ask, what's the basic? How basic is the molecule, and how lipophilic is it? And that will probably get you 9/10 of the way to understanding why that compound is causing that effect. Thinking about secondary pharmacology, we have, there's a lot of companies are doing these broad ligand profiling. Different companies have slight variations on which targets they look at, because a lot of it is based on, I guess, historical experience with certain targets. But there's a lot of publications out there looking at machine learning and using machine learning to predict off-target activity, both of large molecules and small molecules. So obviously, like we would like to think that our antibodies are, large molecule approaches are specific for the targets that they have. But quite often, particularly with things like antisense oligonucleotides, they do hit other targets, and so we have to understand what they are. And so that's part of the approach, is looking at, can we predict what those off targets of large molecules might be. Mentioned a little bit about using machine learning in high content imaging data. We run a number of different assays now, looking at thousands of features within cells that can be used to sort of determine what is happening within that cell. And you can translate those features into sort of a barcode approach, as in the plot on the left-hand side, which then you could start to treat as a barcode, and use that as sort of the machine learning techniques for image analysis to then tease out signals and differentiate. We've used this particular example is looking at a genotoxicity assay that we worked with people at Imperial College, and looking to see if we could differentiate antigen versus clastogen versus the negatives. And so you can see that using feature detection and machine learning, that we were able to identify and correctly classify clastogens versus antigens from a high content assay, which has rapidly increased our ability to predict in these for these two different endpoints. But there are other technologies out there. Some of you may have heard about cell painting, very similar approach to high content screening, using a number of dyes to look at different cellular features, overlaying those dyes sort of simultaneously. You can look at cellular features, you can generate fingerprints of those features, and then start to look at how compounds would cluster in sort of a multi-dimensional space to understand whether you have some degree of toxicity with a compound. Are they clustering things that we know tend to cause certain types of effect? And so in this, there's a paper that's just been published, or is actually, I think it's come out now, using this for PROTACs molecules to identify those PROTACs that would cause mitochondrial toxicity versus those that don't. And so this is a nice piece of work that's used machine learning to look at images. Obviously, multiomics, I'll probably just skip over this. I think multiomics has been around for a long time, but obviously that generates a large number of data points per sample. There are approaches out there looking at using machine learning to predict, in this particular case at the top, predict liver injury. They are now claiming to be able to predict a safe dose, so they look at multiples of your projected human efficacious concentration and determine whether you are likely to see liver injury at a certain threshold above your sort of within a certain threshold of your efficacious concentration. The digital pathology, obviously using machine learning and image recognition. Classical pathology, it's a complex area. I'm not going to say that we solved the problem, but to train a pathologist takes multiple years to train somebody. It takes them a long time to read a slide and has a relatively sort of high error rate on their diagnosis, and obviously we get around that through peer review. But if you could improve upon that using AI and image detection, obviously the complexity hasn't decreased, but you can train algorithms in days, you can get answers in seconds, and typically the error rate in these models in terms of the classification is quite low, at least depending on the type of cell or slide that you're looking at. And these approaches are only getting better as we start to digitize and look at more and more examples of different types of pathologies and tissue types, etc. And as you can imagine, that imaging is becoming increasingly used in pathology, going from sort of the H&E staining type approaches where you look at sort of whole tissue slices, but we can use mass spec imaging now to look at metabolite changes within that tissue slice. We can then start to overlay tissue slices to get three-dimensional images of what is happening within the liver in this particular case, so that we get to a much more detailed level of understanding of safety from the data that we're generating, and obviously then using spatial clustering to try and understand some of that work. So I touched on this at the beginning of the talk, but obviously with the introduction of SEND in 2016 for all NDAs and 2017 for all INDs, we're starting to get an industry-wide standard on raw data submissions. And these offer some attraction now for machine learning, because obviously with standardized data formats, you've overcome about 80% of the work of trying to get the data into a common format for doing machine learning. We get data at the individual level, but also for pooled animals, and so this allows us to get some very complex data sort of for machine learning. But then when you start to look more closely, I'd say SEND is a standard, but it's not quite a standard in the way that we would hope. Some of the fields are still free text, and that introduces errors. So you have to go through unit standardization, correcting spellings, etc., grouping of grouping of similar terms. There's low frequency terms that occur. There are missing rows and missing data within columns that all create challenges for machine learning algorithms. And just as an example, here's one where we looked at male rat heart findings. As you can see, the majority of which were indicated as normal or unremarkable, which again, terminology actually means the same thing. But for the non-pathologists in the audience, it's actually quite challenging sometimes to know whether these things are the same or different. And so when can we merge these frequencies together, and when are they different? And this is where the free text sort of field comes in. Also, some of these could be quite meaningless in terms of their impact on the animal health. So pigmented macrophages, I'm not sure how significant that would be to a pathology readout, for example. And so there's still a little bit of ways to go before we can use SEND in its raw format for machine learning, and there's some data cleansing and groupings that are going to have to be done, I think. So, like John mentioned earlier, we are trying to integrate our machine learning models into the sort of the design, make, test, analyze, so into the early discovery phases where it's appropriate to do it. Obviously, the best way or the most efficient way is to have compounds coming through that don't have safety problems. Similarly, I think we're helping to sort of guide design in a way that helps us to better predict safety and have better PK properties of compounds as early as possible. And obviously, modeling human PK gives us confidence in that preclinical translation at the end to the patient. And so, I think this slide was first put together by Stefan Platz, the head of preclinical safety at AstraZeneca. But I do believe that artificial intelligence isn't going to replace the scientists we have, but those who don't use AI are going to get replaced by those who do, because we're generating so much data that we can't do our jobs effectively without it anymore. And so with that, I'll just finish by acknowledging the many people that have helped with this work, some of which are listed here, and apologies to anybody that I might have missed off. But I do want to thank everybody at AstraZeneca for helping me pull this slide deck together. And with that, I'll stop and ask if there are any questions.
Well, Nigel, great presentation. And before we get into the roundtable, the questions are coming in steadily. Some of them we'll probably deal with in the roundtable, but a couple of specific questions. You commented on the GCNs and the MIT collaboration. Can you expand on that a little bit?
I can. It's a cross-industry consortium with MIT that's being driven by MIT to look at different representations of chemical structures. There's more to it than that. I think the part that we were most interested in was the chemical descriptors, but it's essentially a different approach to representing chemical structures. I can dig out the reference and send it to people if they want. It's not hard. My email address, by the way, is nigel.green@astrazeneca.com. So if people want to ask me questions or want the references, then just let me know. I'll happily provide them.
Okay. And another question, it's posed by multiple people in different forms, right? So your data set, be it for rat or human, this is the PK aspect in the beginning of your talk. Mm-hmm. Is that from internal or for commercial sources? And then the second part of that question is, how much annotation do you really need in terms of assembling that data set? And the third part of the question is, when you're talking about IV PK, that's pretty well established, but oral PK is, of course, very much prone to the formulation. So you could have two different PK sets of data for the same compound with different formulations. So a lot of questions, but just a quick glance.
Okay, I'll try and remember and answer all the questions. So for the rat PK model, that was primarily internal data. We started out looking at primarily sort of the rat sort of cassette dosing type approaches. So the formulations for which were usually fairly standard, I believe, on those. So the formulation part didn't come into effect. I think it has. I agree that formulation can make a big difference, and that's something that we have thought about. We haven't come up with a good solution yet, but we have expanded the model, and at least I think the formulation effects are. The question is how much of a difference does that make compared to the difference between molecules? If mm-hmm. that makes it might.
I get it.
Yeah. So is it just a little bit more noise in the system because of formulation differences, or is it a more significant than we think. And I'm not sure I have the answer to that yet.
And then one aspect related to that question was also how much annotation do you need to assemble these data sets, right?
More is, it depends what you mean by annotation. More is always better. I mean, because you can always then start to ask questions about, do you have over-representation from certain projects? Structural similarity, etc., is always one of those things that we need to think about. But certainly there are certain levels of annotation that you do need. You obviously need to know what the compound is. You have to know what dose was given. You have to know what route of exposure that the compound was given. And you have to know whether it was a male or female rat. And you then have to understand what the parameters were. I think that the formulation is probably another critical piece of information that you would like to have. There are probably additional ones that I am currently blanking on, but I'm sure there will be some that people will consider absolutely vital to understanding the data because they've had experiences with that making a big difference in the past. So I think I'm one of these people that prefers to start with everything and then whittle down to what's important rather than trying to ask the question, what are we missing? So, and then trying to go back and find it because that's a much harder problem. Does that help?
Yeah, this is great. Well, thanks, Nigel. Thanks again for a great presentation. And if the other panelists, aside from the three presenters, and that is John, Rich, and Nigel, and then of course Nick, but if the other panelists, Prashant, Fabio, Beth, could come on video as well, and we can kind of all introduce myself. I don't think I introduced myself. I'm heading the DMPK group with a long-standing interest in machine learning, which actually goes back to my days at Pfizer, where Nigel and I overlapped as well. So why don't we kind of quickly go around introducing ourselves? And if I look at the screen here, maybe Prashant, you can go next.
Thank you. I'm Prashant Desai, currently leading the in silico ADME group at Genentech, and previously I was in a similar role and a project rep role at Eli Lilly.
Beth?
Hi guys, Beth Joshi. Started my career as a chemist, but spent a lot of time in DMPK groups and really have embraced leveraging in silico tools to augment discovery decisions. So I've worked closely with John Sanders back at my time at Merck, but currently now leading the DMPK group for our discovery organization at BMS.
Great, thanks. And then Fabio, another former colleague of mine.
Yes. Hi, everyone. Seems like one large family here. I trained as a chemist, computational chemist, became a DMPK scientist along the way. I built and led the in silico ADME group at Genentech for 8 years, and I recently moved to BMS working with Beth, where essentially I lead the DMPK discovery in the San Diego site.
Thanks. So why don't we begin with a couple of questions. Some can be directly derived from the presentation, but we've got a couple of general questions as well, and we'll probably don't have time to answer all the really interesting questions because this is a great topic. The first question is, to what extent are these models, be it in your own companies or as presented by Rich, John, and Nigel, are they actually used for decision making or just informative, right? So if everyone could kind of comment on that, and that actually reflects on the trust in the models, right? So who wants to go first?
Perhaps I'll start and make an attempt, and others can chime in. I think it depends on the model as to whether you call it decision making or not. I think we have some sort of more guidance for prioritization. So the decisions are never singular data point driven. They're usually multifactorial. And so I think there's a lot of times where the models are used to help prioritize different designs that are brought forward. But questions are usually asked as to why have you made something when it was predicted to be, a potent hERG inhibitor, for example. There might be some questions asked around that. So I think that people will generally not make those compounds moving forward. So I think they are decision making. I think it can vary a little bit depending on the chemist. You touched on confidence in models, and that's certainly something that is an ongoing challenge, I think, within a lot of industry chemistry communities. That I think the golden rule is it can take you 3 months to find a customer to use a model, but only seconds to lose them once they lose trust in the model, right? So it can be quite challenging that if they have a bad experience on one project, they then dismiss models as being useless in general. And so that there can be some, I guess, challenges to overcome in that respect, I think.
Okay.
Maybe I'll jump in and add a little bit more just based on some of my past experiences. I think across the industry, there's quite a bit of data and groups building models on all of those endpoints. But what is really critical within a program team is identifying really what is the critical issue the team is trying to solve for, and then bringing the most appropriate models into those discussions. And where we've had a lot of success, leveraging models for decision making, is really even running a small set of compounds to look at that experimental versus predicted to see how well that SAR is predicted within the global model being used. But really being cognizant to find those key endpoints and then build that weight of evidence such that, within a couple of testing cycles, you're more prospectively leveraging those tools to augment your designs.
Thank you, Beth. Who else wants to chime in?
I would share some.
Okay,
Go ahead,
ahead.
Fabio.
Prashant?
Yeah, so most of my experience, I recently moved to Genentech, so I'm speaking most of my experience at Eli Lilly over the 15 years. And I think what we have noticed there as well is over the time, I think the importance of integrating the modeling scientist along with the project team has really made a big difference in trust in the models. And as more importantly, as Beth said, identifying the right models for the right questions. I think it's a strong partnership between the scientist, those who are either generating data or those who are either using the data, like the project reps, versus those who are building the models. I think it's a key partnership is what has really helped improve the trust in the models. At the same time, I think it's important, as Nigel said, not every model is going to have the same level of impact or same level of performance. So it's important to also make sure that the teams understand what is the overall probability of success with the approach that they are using without the model, and can that be improved by use of the model. So it's not about how accurate the model is, it's about can the model fill a gap that currently exists in the decision-making process, and that has really helped to increase the uptake and trust in the models.
Thank you, Fabio.
Yeah, I'm gonna complete this with some other thoughts, and this is, I don't know if this is your observation as well, but my observation is when it comes to translational science and machine learning, you can easily predict who are the scientists that are going to accept machine learning, just simply looking at, did they accept in vitro modeling? Did they accept the notion of physical chemistry? So I think it's a continuum. If a scientist sees the value of utilizing the modeling way of thinking, they usually adopt machine learning. But if, for example, the view is one of, for example, preclinical PK utilized allometrically to predict human PK, you may be struggling a little bit more, which kind of leads me then to another question for Nigel and John about how do you think of contextualizing your PK predictions in rodent in a human environment so the translation is not lost, and maybe we can get back to that later.
Any final points before we go to the next question? Okay, Nick, why don't you go to the next question?
Yeah, I think Fabio actually asked the next question. Why don't we talk about this translation from preclinical species to humans, and what are the new methods and challenges of that? And I think this is directed during the talk a lot to John and Nigel.
I don't mind starting that one. You know, I think that we have a PPDM group, drug metabolism group, who really specializes in trying to do all that translation really effectively. I think what I presented is a simplification in the sense that obviously those endpoints that we're predicting are of interest, but they're not the only ones that are of interest. And so, you know, through some private conversation with Fabio behind the scenes, of course, there are in vitro considerations and species differences that are important. One of the things that we do is we use Spotfire as a platform to share a lot of different information with teams. And one of the specific things that we do inside that platform, and this varies from project to project, is through the partnership with our PPDM colleagues, we'll deploy early dose predictions based on a combination of in silico, in vitro, and in vivo data. And as better data closer to the end goal of being in a human become available, we will selectively pick those data points to drive those dose predictions effectively. And so I think that there are absolutely cases where we will combine those kinds of data. It's never as simple as just using a simple rat prediction, but allometry is a pretty key assumption in most of our work.
And I'll maybe add, John, just because I was sort of involved in a lot of this, you know, we used to use, well, we still do, a lot of heat maps too, and partnering endpoints in close proximity, because I think a lot of times the modeling will help really inform maybe key studies you want to bring forward to test some assumptions. So John mentioned, you know, allometric scaling being a first very early approach. But if there, you know, are modeled endpoints that make you think a little bit differently, that's been really insightful for us to really work with teams and, you know, refine those assumptions, because ultimately we're not, you know, designing drugs for rodents. We're really doing that for clinical patients. And the sooner we understand these things, the better.
So I'll perhaps just chime in. I think certainly we use Spotfire as well to communicate back the performance of models. Whenever we retrain with models, we always look at how well did the previous version of the model predict that experimental data coming out of the assay. And that data is available for all project teams. They can, you know, it's labeled by project team, so they can actually go in and look at how well the model was doing for their particular project and their compound. So they can get a very quick understanding of how well that model is performing. I do struggle a little bit though, that I think the experimental data that we generate isn't quite as good as the chemists like to believe it is. That they're increasingly wanting to say that the experimental data is the ground truth, when it's an N of 1 from a first batch of a compound that they've just synthesized, so the first time they've synthesized it, and it's sort of a human liver microsome wave 1 assay is it's not necessarily the quite the cleanest of data that they might expect. And so I think we've had to do a lot of education around what's the experimental variability of the data that they have, so that they get an understanding that there are going to be differences between a prediction from an in silico approach and a point value that they get from a single test in a HLM assay, for example. And I think once they start to understand that, you get a much better sort of reasoning with the chemists about the performance of the model. It's not that one is right and one is wrong, it's that they're in the same ballpark, right? And that's the important thing.
Yeah, I think, Nigel, that's an important point. You always got to think about variability in anything that you predict. And in some cases, for our in vitro models, the variability it actually really affects the output. And in some cases, the output of the computational model actually begins to approach the experimental variability. And that's, of course, the limit of where you can go with the models. I'm going to combine a couple of questions into one question. And again, we can kind of go around. There were questions about when do you go with a local versus a global model. And related to that question, and I understand it's a hard question to answer, how many data points do you really need? And so whoever wants to comment on that.
How long is a string? Is the answer to that. It really depends on the endpoint, doesn't it? And also the local versus global. I mean, that's a great question. I think to some extent that depends on how generalizable is the problem and how good is the model at that time point. Obviously, if you can have comparable performance with a global model, you want to use that because there is more likelihood that that will extrapolate beyond the current space. But I'm interested in hearing other thoughts.
Rich, can you look at it from a large molecule perspective as well?
Well, I think for us, the answer actually goes back to answering a question of, how do you get people to engage with these models and what are some of the pitfalls there? I think for us, we are really pushing towards a global model. I don't know if it's the right answer, but that's where we're putting all of our chips. And there, the idea is just, if we're going to avoid a late stage, we know that we need a huge concentration of this molecule in some very late stage, almost formulation problem. Well, if it's a very simple thing to encode, surface patch charges and things, in the early stage when we're also looking at affinity, well, those surface residues are going to interact with the same surface places where we want to engineer stability and affinity. So we need to put those really late stage things and those early things together, because otherwise those oracles are fighting it out for the same real estate. So I do think that a global model is the right thing, but then it leads to this very scary thing where you say, hey, we're going to make you, these are the molecules. And you get really instant feedback for some of the first things you assay, like expression and affinity, or in the small molecule case, potency. And then you wait a super long time for the other parts that you're hoping are hiding there or already dealt with in some way. And I think when we saw John talk, he showed these distributions of what people make, and you have to do some detective work to see whether or not it's really having that impact. That was really, really great, John. But for us, I think that we do embrace these global models. And then as far as the number of data points goes, I think it differs depending on the quality. We heard a little bit there, but there's another, this is the scary thing. What if you use active learning and algorithms to select optimal things to test? Can you get to the point where you're actually honing in on the right answer and therefore becoming very data efficient at the end of the process after many iterations? But that's really scary, because all of these we're in such high dimensional spaces that are we just really jamming a local minima, or are we actually extrapolating and doing cool things? And that's this super active debate, not just in our field but across machine learning. But I do think that active learning can help be more data efficient, and I do think the global is the right answer. But I'm also just, terrified of, both of those answers. So,
Thanks, Rich. Someone else wanted to say something about this topic before we go to the next question.
I think
go ahead,
sorry.
Nigel. Go ahead, Nigel.
I would agree with Fabio on this, that I think the global models you've got some degree of comfort that you're going to be able to go beyond sort of or extrapolate a little bit, that it's not going to fall over dramatically as you go beyond the chemical space. I think if you can guarantee that you're staying within the confines of a local model, which is challenging to do computationally to come up with the applicability domain. I think there's been more papers and arguments about how to define those things than I care to mention. But if you can define an applicability domain for a local model and know when you're going too far outside it, then perhaps they do work better. But I'm not convinced, to be perfectly honest. I think you just get similar performance from smaller training sets, basically.
Great. Nick, why don't we go to the next question to keep it going?
Can I jump in one
think,
thing about this
go
before we move on? I think there's also a pragmatic reason to focus on global models. So I happen to believe in global models based on a lot of internal data that I've seen. Bob Sheridan's work is pretty influential for me in that regard. But I think a lot of the modelers that I've seen who build local models for their teams often will want to build a local model because they think that it's going to be more performative. And I think one of the risks in that is you can spend a lot of resources building a local model and putting it side by side with a global model, and then you have potentially a few dozen local ADME models when you've got a global model that's launched. If they don't perform any better, you've then effectively increased your workload by an order of magnitude, and the maintenance burden that goes up with that, and also the ability for an end user to pick the right model for their own needs becomes more complex. And so I tend to prefer sort of a pragmatic, simple approach that also happens to be pretty effective. And as long as you're updating your models with regularity, I think that you can incorporate all that local data on a very reasonable time frame.
Nick,
Yes, I see. Another, I think, interesting question from the audience. They're asking, what are the main challenges that need to be overcome to enable cross-department collaboration on the different in silico models, from chemistry to protein engineering to DMPK, then PK/PD and pharmacometrics? Is there a need or advantage to integrate these different in silico platforms, or do you think they should remain semi-separate?
I can go. I think it's really, really very important question. I think the question really highlights some of the challenges that many of us have seen, that even with the success of individual models from different areas, I think the teams that are benefiting the most are the ones that are able to actually integrate models from different area of science. So potency, for example, ADME, for example, even formulation related models. And I think one of the solution is to really build more deliberate connections between these different functions, both at the project team levels, but also quote unquote behind the scene, where the modeling groups should be encouraged to work together, because a lot of time the techniques that these groups use are very similar. It's just the approach may be different depending on what you're trying to predict. And I think having some more cross-functional understanding of the science of different areas would also help. In my experience, I have noticed that potency SAR typically would behave differently than, let's say, an ADME SAR, mainly being, potency SAR likely to have more activity cliffs, where a small change in structure could make a big difference in potency, which is not very common when you look at the ADME endpoint. So I think having that kind of sharing and exchanging that understanding of the different data side to different sciences can really help integrate the models together, and that would be the right way to actually get the most out of these models across the entire project teams.
Maybe I'll jump in really quick and add a little bit to what Prashant shared. I think there is often an underlying theme across some of the endpoints where potency and some of the DMPK endpoints and maybe even some of the safety endpoints have an underpinning of lipophilicity being impactful in how those parameters move. And so, looking at that in isolation, you may be prone to kind of setting up arbitrary cutoffs that, end up being maybe negative, maybe positive for a DMPK endpoint, but negative from a potency perspective. And so, I've always been a big advocate of bringing that cross-functional team together and really deciding what are the key things that we want to improve or directionally enrich towards as we're trying to learn about, what potency and interactions give us, favorable enthalpic binding, and how does that track with some of the DMPK endpoints that maybe, orthogonally influenced. And so trying to do those in isolation, you miss the touch points between the different functions. So in all of my experience so far, it's been those teams that really come together, align, and understand how those move help teams to kind of build these ratios or MPO approaches to try and define success.
Anyone else wants to comment on that question?
Yeah, perhaps I'll just chime in on what Beth just said. I think you're absolutely right that there's a lot of interdependencies in all of this space, and so it's important to work together. Certainly, we've noticed, particularly in safety, that things like lipophilicity obviously lead to off-target binding effects in secondary pharmacology assays, which then lead to things like cytotoxicity because you're just hitting a whole bunch of different things. Then you see cytotoxicity in your liver assays, you see them in your cardiomyocytes as well. And so it starts lighting up all these flags in all of your assays. And the individual target organ specialists that are looking just at their assays are not asking the question, is there more to it than that? And so I think we've noticed that it's very important for us to bring, ironically, medicinal chemists to the table with the toxicologists to actually talk about what's the holistic view of the molecule. Part of my group, I have 4 medicinal chemists now that actually sort of act as that liaison between the chemistry groups and the safety specialists, so that we get that dialogue working correctly. Because that has actually been a part of the problem, is that the safety organization sort of was seeing cytotoxicity as a bad thing, but it was being driven from a lipophilic molecule that was just hitting a whole bunch of kinases. And it's like, well, yeah, tell me something I don't know, kind of thing. So I think you're right. There's a big drive to start to bring a multidisciplinary team together to look at these issues, and with that we start to understand more about what's driving a lot of what we see in the assays that we use.
Thanks, Nigel. And the next question is probably going to be the last question, because we are beginning to approach noon East Coast time. And that is, what challenges do you see? And in light of those challenges, what does the future hold? And I'll throw in one thing based on what Prashant and Beth said as well. In some areas, we've made tremendous progress in the prediction of ADME properties, still some limitations, but overall we're doing a very solid job, right? But predicting potency, to me, that's really the holy grail. And as Prashant said, that's still very challenging, right? So again, what are the challenges and what does the future hold? Who wants to go first?
Well, I actually want to take this question, narrow it down to another question and direct it to Rich, because I'm very interested in the active learning aspect of that. But I do have kind of something I need to sort out in my mind, and I'm interested in your opinion, Rich. Active learning, it's successful if at the beginning of the journey we know the relevant questions, I think. And I'm finding during research projects that a lot of the relevant questions are emerging as we go. How do you frame that?
Yeah, so that's one, right? So that's one thing. The models, the research questions, and also the way people use the tools are changing as we go. Another thing is there's lots of cases where if you have a no-go at this, there's no point measuring these other things downstream. So applying active learning in this area is a lot more challenging than other things like images and maybe speech, I think. So one of the things, so we've done a couple things. One thing that was really quite brilliant, we had a student who came out of Andrew Gordon Wilson's lab who figured out a way to put together the active learning components into what we call property DAG, or directed acyclic graph. And so then essentially we can talk to people who know what they're doing, like Beth, and we can say which pairs of people are trying to change molecules with similar methods or concerned about the same properties. They might be at different stages of the pipeline, but which pairs of people are working on related properties, and which pairs of groups or subsets of groups are actually properly upstream and downstream from each other. And then the other thing is you can use machine learning to say what part of the input space is being modified, right? Are these things that are changing? You know, and you can use GradCam and you can use ways of seeing what parts of feature spaces are important to see how methods are competing for the same real estate, you know, and in this imaginary space of machine learning models. So when we do that, we're able to sort of put together essentially component active learning modules that have a hierarchy, right? And so for us, you know, I think for small molecule, can you make it? For us, it's does it express, right? Those have to be, you know, overarching. Then you can descend into these other things. And the model needs that hierarchy, because otherwise it's imagining all kinds of things that you can't make one way or another, and it's thinking that those are negatives, and they're not. They're just irrelevant in a way that's sort of mathematically different. So for us, switching to this hierarchical framework, it's still pretty new, but it's had a few iterations and it's shown its mettle so far. That was really important. Another thing is to step back and say the model's changing out from under our feet, including how people use them, does make it a lot harder. And so there's some cases where we might just have to reset the system and say, no, you know, we're now in a completely new regime in terms of how people are using the tools, and the experimental technologies are much higher throughput. So we have to hit the reset button, put it all together as one big data set, and then start the process of active learning over again. I think every once in a while, we just have to declare, you know, tactical retreat, smush the data together and restart the active learning process. Fabio, I'm not sure if I answered your question, but maybe I just put all my worries on the table in a different way. But that's how we're approaching those things right now at Genentech. And most of what I'm talking about is also in the large molecule space. And so I would have to think through things with this group to think the right hierarchy for the SMDD space.
Any final comments before I hand it back to Nick and James? All right. Well, thanks everyone for the presentations and a great discussion. We probably could have gone on for another couple of hours, but time's up. Nick, James.
Yeah, are you able to see my screen?
Yes.
1 second. Looks like a little bit of a glitch going on. Okay, I can't really see my PowerPoint slide, but can you just see the PowerPoint slide?
Yeah.
It looks good. Yeah.
Okay. Yeah, I didn't know if James was able to show his video, but I just wanted to end with some concluding remarks, and thank you everyone for being a part of this virtual IQ workshop. I think we accomplished our goal of starting a robust discussion about the application of AI and machine learning across all the different stages of drug development, and I think we've identified some good practices, but also some challenges and future directions for this work. I did want to just remind everyone again that if you missed any of the sessions, that the recordings and the slides will be posted online with a public link after this workshop is over. And I know that we weren't able to get to all of the questions that were submitted today, and those will also be submitted to the presenters offline, and hopefully they are able to get back to you. I know we're pretty much out of time, but I wanted to really thank everyone that was involved with this, and give a special thanks to Maya Leah Marshall, who helped the organization of this event, but also for the other IQ Consortium workshop organizers. I wanted to thank all of our speakers that had to work on a semi-tight deadline to help us create this program. and of course all of our panelists that have contributed to this discussion that I hope we continue going forwards. And finally, I wanted to give a special thanks to our IQ group working members. I apologize if I missed someone in this list, but these are all the individuals that have been working over this past year to work on an FDA scientific exchange last year, and this year a virtual workshop. So I think this is a starting point. I think that we will continue the conversations on this topic going forward.
Yeah, thank you everyone for this, for your contributions and making this a very successful event.
Thank you very much.
Thanks, everyone.
Thank you.
Thank you.
Bye, everyone.
Thank
Bye.
you.
Bye.
Bye.